This recording is an introduction to sensory pathways, in particular the somatic senses, but I do want to discuss general differences between things such as special senses and general senses. I'll also go over a basic structure of sensory receptors, a process called transduction and adaptation. We'll look at some basic sensory receptors, and kind of some basic introduction to sensory pathways. In class, we'll talk about specific pathways, such as like pain, how pain is transmitted, things like temperature and those type of things. So just kind of like as an overview, I want you to realize the difference between special senses and your general senses. Your special senses include vision, hearing and equilibrium, smell, and taste. And so those receptors are very highly specialized receptors that we find within the head. And those information we'd always talked about were carried along cranial nerves. So we'll actually review a little bit of the cranial nerves too. Now, anything other than the special senses encompasses general senses. Okay, so anything like touch and pain, those are pressure, those would be considered general senses. Now, we also can distinguish senses and break them down into somatic versus visceral. So somatic senses is what we're primarily going to be discussing in this lecture, is the things coming from, say, your skin, like touch and pressure, pain receptors. It carries information in regards to information coming from your joints and muscles. The visceral senses include things coming from your visceral organs or your internal organs. So say, for example, your stomach gets stretched. That stretching of that stomach is carried along what we call visceral sensory neurons. Now, what I want to do, though, is kind of break down the different types of fibers that carry the sensory information. Now, remember, we've had discussed the difference between afferent neurons and efferent neurons. Afferent neurons are sensory neurons, and the fibers that carry that information referred to as afferent fibers or afferents. Now, we have fibers that carry, or we refer to as special somatic afferents, so it has to do with special sense. And the somatic here is things in regards to your vision, hearing, and equilibrium. They have, they're carried along these special somatic afferents. So as a kind of a rule, think somatic things from the outside, like you have to hear something outside. You're looking at someone from the outside. So they're carrying that type of information. Now, you should remember the cranial nerves that contain those specific fibers. Hearing and equilibrium is associated with cranial nerve 8, which is your vestibulocochlear nerve. That nerve is purely sensory, and those are the only type of fibers that you'll find within that nerve. Vision is carried along cranial nerve number 2, which is your optic nerve. That again is purely sensory, and it only carries that particular type of afferent fibers. Now, the special visceral afferents carry information in regards to both taste and smell. Now, smell is the cranial nerve associated with smell is your olfactory nerve, which is cranial nerve number 1. And again, that is a purely sensory nerve, so that's the only type of fibers that you'll find within that nerve. Now, taste, that information is carried along one of 3 different cranial nerves. Now, unfortunately, those cranial nerves are mixed nerves, so the special visceral afferents aren't the only type of fibers that you'll find within those nerves. So, for example, because they're mixed, they have motor fibers or efferents, and that's something that can be discussed at a later time. Now, taste could be associated or carried along cranial nerve number 7, facial, 9, glossopharyngeal, or 10, the vagus. Okay, so they will have those type of fibers along with some other type of fibers. Now, then we can also, instead of talking about special afferents, we can discuss general afferents, and again, we can divide them into somatic and visceral. So I'd mentioned before, the somatic senses, things or information is coming from your joints and your muscles and your skin. So various information regards to temperature and pain and touch. That's what we're going to be talking a lot about in this particular lecture. The special visceral afferents, that information is coming from your internal body organs, or could be also glands and blood vessels. They're going to be important, say, in a lot of when we talk about visceral reflexes, so things like blood pressure regulation, they would be important in sending the sensory information to the central nervous system in regards to those reflexes. So what I want to do next is talk about this whole process of integration, how the central nervous system is going to be important in the integration of sensory information. Now we have to break this, introduce you to some new terminology, is sensory information is carried to the central nervous system, and we're going to refer to that kind of raw form in which receptors send the information to the brain, and we're going to refer to it as a sensation. Now, the central nervous system is somehow going to be aware of that sensation, or they're going to interpret that information. So the awareness of that sensation, or how the brain interprets it, is, so we're going to say awareness of the sensation. We actually refer to that as a perception. So I have a picture here to kind of give you examples. Is you're watching a baseball game, and you're hearing like the ball hit the bat. You're hearing the crowd roar. So you have a lot of information that that's being processed. Is that visual information, the sound waves, are carried along these sensory nerves as sensations. So it's kind of like a raw type of thing. But the brain is then going to process it. What do I need to do? Do I need to boo? Do I need to clap? Do I need to stand up? So how your brain interprets that information is referred to as perception. So I'll give you another example between sensation and perception. Is just light itself is a sensation, but your brain interpreting it as blue, a color, like red, that's perception. Or another example would be is a smell. That's a sensation, but what you interpret that smell as, is it a certain type of perfume? Is it bacon cooking? That type of stuff. That's a perception. So one thing to keep in mind, though, you can have sensations without perceptions. So if the brain does not process the information, it's never perceived. So that can actually happen. So let's talk about next is kind of how we're going to kind of get that information ultimately up into the central nervous system. And so let's first talk about is sensory receptors. So these are the receptors that are responsible for responding to certain type of stimuli. And when we look at sensory receptors, not all of them are created equally. They're all a little bit different structurally. We have ones that we call free nerve endings, or we call naked nerve endings. We have ones that you see like right here that have enclosed. They have some connective tissue wrapper around it, and you have some ones that are very specialized. Now, as a good reminder, kind of showing you these sensory neurons, is what you see here. These are the blue, are axons. Over here, this is a dendrite. These are dendrites. You see the dendrite here and there. And so you got, on that free nerve ending, those dendrites are actually the specialized sensory receptors. That information is going to be carried along the axon, eventually up to another neuron, and we'll talk about that later. And here again you see is the soma, or the cell body. So remember, this is what you see here is that typical unipolar neuron that we've seen previously. Now the enclosed nerve endings, how they're different is you see they're encapsulated. So you have a lot of this connective tissue capsule, which with the kind of distinct, the actual reason for, or an advantage of having that is it actually enhances their sensitivity or how selective they are. The ones that have the naked nerve endings are not as structurally specialized. They're not as necessarily as selective or sensitive. Now the specialized receptors are very specialized. They often have, you'll see here, is it can release neurotransmitters. So they're a lot more complex. But you'll also notice that we have some, on the comparison here is we have some sensory neurons that may be myelinated, which you see here. Here's myelin. And you may have ones that, like this example I drew here, is unmyelinated. So remember the difference is if you have things that are myelinated, it can carry information quicker than if something is unmyelinated. Now I want to give you some examples of specific sensory receptors in regards to their structure. So the ones that have these free nerve endings that we're going to actually talk about is, so here, the free nerve endings, the dendrites are the sensory receptor. And again, on this example, though, you see in this particular example, it actually is myelinated. But we're going to be looking at nociceptors. And there's two different ways. You can spell it with or without this O. Nocioceptors and nociceptors, they are involved in pain detection. Thermoreceptors are involved in temperature. Just going to abbreviate temperature, like cold and hot. Other example would be, let's see, your tickle, itch, receptors. We're actually not going to talk about those, but we will talk about baroreceptors. These are involved in pressure, detecting of pressure, and things like blood pressure, that type of stuff. But these ones we'll talk about a little bit the most. The ones examples that are encapsulated or lamellated, they have these enclosed nerve endings. We've talked about these previously. So I'll give you an example. Pacinian corpuscles. Again, we'll review them again. And Meissner's corpuscles. So remember, Pacinian corpuscles detect vibration and pressure, like someone pressing down on your skin. Meissner's corpuscles, that's in light touch and dynamic deformation of the skin. And we'll introduce you a couple other types of receptors that have encapsulated nerve endings. The specialized ones, these ones are involved in your special senses. So remember, your rods and your cones are involved in vision. These are just generically referred to as photoreceptors. Can't spell. You have your two receptors that are involved in equilibrium. The macula that are found in the vestibule, remember utricle and saccule are part of the vestibule, though that's important in static equilibrium. You have your crista ampullaris, which is involved in dynamic equilibrium. Those were found in the semicircular canals within the ampulla. So here's your semicircular canals here. So remember, this is the vestibule, and these are your semicircular canals. Now the smell is going to be, or your olfactory receptors, those are specialized. You have your gustatory receptors. They're involved in taste. Now, I forgot to put the picture, I can come back here, is within the cochlea, you have your organ of Corti, which is involved in hearing. So these are all very specialized type of receptors. Okay, I just did that mainly as a review for lab. So you have these different receptors. Their job is to respond to certain stimuli in the environment. Now we have to somehow get that information and figure out a way to carry that information to the central nervous system so we can interpret it. And the thing is, before we do that, still I want to talk about a little bit this thing called receptive fields. is you have these different receptors that have what we refer to as different receptive fields. A receptive field is an area within a receptor that can detect a stimulus. So here's a receptor. It's like this is the area that it can detect any type of stimulus. But you have some types of receptors have very small receptive fields, and you have ones that have very large receptive fields. The difference between that is if you have a very large receptive field, your ability to discern the specific location of that stimulus is going to be poorer. So you have a much smaller receptive field, you have a better idea of where that stimulus is directly coming from. You can localize it. So put it like the smaller the receptive field, the better you can localize it. And I'll give you an example. Something that could be done in lab if you wanted to is you can do something called a 2-point discrimination test, where they take these specialized little calipers and they keep them really close together, and you're going to place it on a particular body part, and the subject's not going to look at it, but they're going to tell you, do I feel 1 or 2 points? And you start real small, it's like, okay, I feel 1 point, and you keep slowly opening that up like you see here. You move it like a millimeter at a time, and the subject's going to say, I still feel 1 point. And they keep going. Oh, still feel 1 point. The point where like, oh, I can distinctly feel 2 points, then they're going to say, okay, that's where, how well they can discriminate, where that sensory information is coming from. What you'll notice is things like your fingertips. You can distinguish between 2 points very early on. If you do this in the back on your, even on the back of your hand or the back of your shoulder on your back, you can't discern where the 2 points are until they're much, much further apart. So your fingertips have the ability to detect 2 points a lot better than, say, when you look at your sensory receptors in the back. So they have much smaller receptive fields in your fingertips versus the back. Now here, this is where I want to talk about how we're going to get that stimulus converted into some sort of information that we can eventually carry it to an area for interpretation. So we refer to as transduction. So you're going to change that stimulus into a certain type of potential, and we're going to call it a generator potential or an action potential. And what you'll see is kind of part of this is a little review from some other stuff that we've talked about, is you have a stimulus, and what you'll notice is you have to have a certain threshold. It must be intense enough to be able to generate an action potential. And so what you'll notice is we're going to refer to is the receptor potential is a graded potential. So remember, we have discussed graded potentials before. Graded potentials are not action potentials, but they can lead to an action potential if they're excitatory. So you notice go from here to here, the intensity increased. This one here wasn't enough to reach threshold. Okay, so you didn't see an action potential. But this one was a great enough stimuli that we started to see action potentials. So these ones are referred to as receptor potentials. So they're graded potentials. When we're talking about sensory information, we refer to the graded potentials as being graded or receptor potentials. If you have enough of a stimulus intensity that you can reach threshold, you will have an action potential. And the action potentials referred to as generator potentials. And so you'll see the action potentials then will be transmitted along the axon. Eventually to the end, you see here, we'll get release of neurotransmitters, and then we'll hopefully generate another action potential on the next neuron that's going to be carrying that information along the central nervous system. Now what you're going to notice, though it's very important that you notice this, is if you compare these two pictures, this one and this one. Remember, action potentials are all or none. So all the action potentials are going to appear the same. How does your nervous system interpret differences in intensity? So if I was pressing down on your hand, How does your nervous system know that I'm applying greater pressure? It comes down to the frequency of action potentials. So if you compare the left to the right, is the differences in intensity is how we're going to interpret it is the frequency of the action potentials changed. How they looked are the same, it's all or none, but how your nervous system is going to interpret the strength of the stimulus is by the frequency of action potentials. That's how we decode information. Okay, it's going to be frequency of the action potentials. Okay, so you'll notice again, we'll go as you have a stimulus that will lead to a graded potential, or referred to as a receptor potential. If it's enough of an excitatory stimulus, enough to reach threshold, we will have a generator potential or an action potential, which will be propagated along that axon. And you notice on the picture, that is a myelinated axon, so it's going to jump from node to node, and eventually we're going to somehow have to transduce or transducing information along the axon, going to have to get it to the next neuron. It's going to involve some some release of neurotransmitters at the synapse, and will generate another action potential along the next neuron. Now, where eventually that information is carried to the central nervous system, and your central nervous system is going to interpret that information. So I do want to do a little review of some information that we've talked about before, is how are you going to perceive that information? So remember, you have a sensation, but it hopefully leads to perception. So it's going to be going to either somewhere in the cerebellum, or say somewhere in the cerebral cortex, where we can process certain types of information. Now, the primary somatosensory cortex is a region in the parietal lobe, and so it's right here. It's in the postcentral gyrus. Remember, that is going to interpret information in regards to touch and pressure and pain, the somatic sensories. So the somatic senses that we're going to talk about is going to be processed in this specific region in the brain. So it's in the parietal lobe. And that primary somatosensory cortex is located in the post-central gyrus. Now, the visual cortex, which you see in blue back here, is found in the occipital lobe. And that's going to be processing visual information. So information coming from the optic nerve, that's going to be processed in the occipital lobe, in the visual cortex. The gustatory cortex, can't really see it here. We have to pull the temporal lobe out, and so you can see the insula. So the gustatory cortex is in the insula. That's going to interpret taste, information in regards to taste. So information coming from facial, glossopharyngeal, vagus nerve. The information in regards to smell is processed in the olfactory cortex, which is you're going to find in your temporal lobe. Okay, so olfactory cortex is in the temporal lobe. The auditory cortex, missed the auditory cortex, I didn't picture. The auditory cortex is also going to be found in the temporal lobe, so that's for hearing. So information from the vestibulocochlear nerve in regards to hearing, specific cochlear branch of the vestibulocochlear nerve. Now, information in regards to equilibrium and subconscious proprioception does not reach conscious awareness, that does not go up to the cerebrum. It actually goes to the cerebellum. So it's within the cerebellar cortex, within the cerebellum. Okay? Now, I have to mention that sometimes some information is going to be processed within the spinal cord. Those would be involved in like reflexes and stuff. Like if you remember the knee jerk reflex, someone taps the knee. That is going to be processed within the central nervous system, yes. And spinal cord is part of the central nervous system. But at some point you are aware that someone tapped your knee, and that will go up eventually to the cerebrum. but there is some information that's processed in the spinal cord, and if it doesn't go up to the cerebrum, you're not aware of it. Okay, so just remember as a rule, if something doesn't eventually go up to the cerebrum, you're not necessarily consciously aware of it, but your body is still interpreting that information. Now, some sensory receptors can adapt. So I do want to talk about this thing called adaptation. Is we have receptors that we refer to as slowly adapting, and we have ones that refer to as rapidly adapting. Adaptation, that term adaptation, is where you have reduction in sensitivity of a receptor despite the continuous presence of a stimulus. Somehow the receptor membranes become less responsive, the receptor potentials decline, and they can, the frequency can decline, or they eventually can stop. And we have some receptors that adapt relatively slowly or not at all, and we have ones that actually adapt very quickly. So if you look in the left picture, left and right, actually you have a continuous stimulus in both of them here. But with the slowly adapting, you still have receptor potentials. But in a rapidly adapting, a point that stopped, there's no receptor potentials, so that information is no longer being sent to the central nervous system. So I'm just going to give you an example of something that rapidly adapts that you may be aware of, is smell. If you ever walk into a room and somebody has some perfume on, you initially smell that perfume. After a while, you kind of become accustomed to it. You don't really notice it as much, or people don't realize that you adapt like your dog. You're used to the smell of your dog, it doesn't bother you, but other people come in your house and they're like, wait, it smells like dog. They're going to say, it smells like dog. But eventually they kind of get used to it as they stay in there in the presence of the dog. So smell, the olfactory receptors actually rapidly adapt. Temperature receptors do too. Slowly adapting receptors include things like pain receptors. You don't want them to adapt. You want to always be aware that something is potentially harming you. So they should adapt. Now, receptors, we can also refer to them as, besides being slowly adapting or rapidly adapting, we can use the term tonic or phasic receptors. So the picture on the left shows you a tonic receptor. So tonic receptors are don't adapt, or adapt very slowly. These guys are going to be important in information that's about a stimulus is going to be very important, like pain or stretch, things that could be potentially harmful to you. Phasic receptors rapidly adapt, so they turn off. So they're important for only when there's a change in the stimulus, or the intensity, or more important, like rate of change of the stimulus. So like touch. So when something first touches you, you know it, but then unless they remove their finger from you, lots of times eventually you kind of get used to it and you don't really notice. Like your clothes. You don't notice that your clothes are on until I said that to you. You're like, oh, I can feel my clothes on you. Those would be phasic receptors. So again, what you'll see on the picture, the tonic receptors, you have continuous stimulus, but you'll notice is that receptor potential, that graded potential is decreasing. And you notice here with the action potentials, or the generator potentials, the frequency is getting further and further apart, but not to a point where, in this case, it stopped. However, with the phasic receptors, you saw in the initial stimulus, you saw that receptor potential, and you got some action potentials, but then after a while, they stopped. So tonic receptors, you can also kind of interchange them as they happen to be slowly adapting receptors. They're always on. Phasic receptors, they're just like they're on. They adapt so quickly. They're just kind of like they're either on or they're off. Okay, so they, if something's continuous stimulus, they kind of like, yeah, I'll just wait until something changes. So those are phasic receptors. So what we're going to be doing is, what we'll be doing in class is we're going to be looking at specific types of pathways, and we're going to classify them by this nature of the stimulus, the different types of receptors involved, and how the information eventually is carried up to the central nervous system, and ultimately how we're going to process it. So we're going to just introduce you to kind of the organization of these pathways. We're going to break things down into different neurons. We refer to as a first, second, in some instances we have third order neurons. The first order neurons are your sensory neurons. So when we've talked about sensory neurons, that's really what I've been referred to as. So first order neurons are your sensory or your afferent neurons. Okay? Their cell bodies, remember, are going to be located, you'll notice right here, either in the dorsal root ganglion, okay, I'm going to just abbreviate, DRG, dorsal root ganglion, or if this has to do with some of the cranial nerves, they'll be in cranial ganglia. Okay, so that's where their cell bodies are going to be. They carry their information towards the central nervous system. On this picture, see it going to the spinal cord. The second order neuron is an interneuron. So the cell bodies for these second order neurons are going to be located in the dorsal gray horn. You see back here. So they're in the dorsal gray horn of the spinal cord. So dorsal gray horn of the spinal cord, or within the medullary nuclei, within the medulla oblongata. Depends on the particular pathway, and we'll make that distinction later. So it's in the dorsal gray horn of the spinal cord or in medullary nuclei. So there are nuclei within the medulla oblongata. The second order neurons, now the second order neurons will transmit the impulses either to the thalamus. It'll go along to the thalamus if the information is to reach conscious awareness, because that eventually will take it up to the primary somatosensory cortex. Or it can transmit information to here, the cerebellum. So some of the pathways that we look at in which it doesn't reach conscious awareness, so we refer to as the spinal cerebellar pathways, they go to the cerebellum. You see, in some instances, you're going to see, depending on which pathway we're talking about, is you will get decussation, crossing over. This is why right side of your brain is interpreting information from the opposite side of your body and vice versa because of crossing over. And that will be important when we talk about those pathways, is knowing where something crosses over, because that'll be important clinically. So we'll talk about that a little bit later. Now, third order neurons are only involved in the pathways in which the information reaches conscious awareness. So it has to go to the primary somatosensory cortex. So the third order neuron will be in the thalamus. The thalamus is then going to direct the information to the appropriate location within the cerebral cortex, and it knows exactly where to go. If it's coming from the leg, it knows exactly where in the cerebral cortex that information is going to go to. The thalamus will also be directing information, of course, say some of the special senses, but we're going to talk about the somatic senses, so we're all ultimately going to go to the primary somatosensory cortex. Now, the primary somatosensory cortex is actually mapped out, and it's referred to as a homunculus. And we have what we call a sensory homunculus and a motor homunculus. So we're just going to discuss right now a sensory homunculus. A homunculus is kind of a picture representation of the cerebral cortex, and specifically for sensory homunculus, it's a pictorial representation of the primary somatosensory cortex. On this picture, you're only seeing one, but we have them on both sides. What you'll notice is they use this term somatotopy or somatotropy. It's a particular area of the body correspond to a specific point in the brain. So, for example, information from your feet. So if I touch something on your foot, that information is interpreted within a specific point with that primary somatosensory cortex. And you'll notice it's right here, kind of closer to where that longitudinal fissure is. And what you'll notice is the picture, and then the one picture on the left is kind of always homunculus means little man. To me, I call it creepy little man because they look creepy. Is the way that it's drawn is the different areas of the body are going to be you get these kind of distorted human body. You'll notice you have very large hands on this picture, very large lips, face, and what that corresponds to is the area or the amount of cerebral cortex devoted to a given body region is not proportional to the body's region's surface area or volume, but rather how innervated that region is. So you have a lot of sensory innervation of the hands and the lips and the tongue and the ear. So you'll see disproportionate on the picture, the actual map here, is you see a very large hand, very large lips. And the thing is, these maps can change depending on experience. So if you do a lot of things with other stuff, it can actually be altered somewhat. Also, what I want you to notice is that this is very orderly. The map is, and there's a rule, we call it medial to kind of lateral rule. Information coming from the lower part of your body is going to be going to areas more medial. Okay, so you see it here. As we ascend, the further up that information is coming from, that's going to be sent to areas more lateral. Okay, so you'll notice the feet's along the longitudinal fissure, and as we're getting closer to that not central sulcus, the lateral sulcus, you're starting to get more to the head. But here, like right deep in the lateral sulcus, you're getting in more in the mouth area, and the tongue, and the pharynx inside the head. But that's kind of as a general rule. So, for example, someone came up with this at some point, is if you have open brain surgery and the person is kept conscious, is if they stimulate, say, this particular right here, part of the primary somatosensory cortex, and we'll say this was on the right side, the person is actually going to feel like someone touched them in their butt, but it's going to be the left side, because what we're going to talk about is the right side of the brain interprets information from the left side of the body. So if someone actually stimulated that area, they're going to think that someone actually touched them on the left buttock. Okay, so they can that's when they could do this. They could stimulate certain areas, and people are going to think, someone touched me here, someone touched me here. So it's kind of a cool type of thing that they do. Now, we're going to do, I'm going to just introduce you to the tracts that we find within the spinal cord that's going to be carrying certain types of information. And we're going to break those tracts up into, we have two that are found in the posterior white column, in the blue, which you'll notice right here. Or these tracts actually cross over at the level of the medulla oblongata. The two tracts are, we have fasciculus gracilis tract, you see on this side here, and then on over here and right here, that's the fasciculus cuneatus tract. We'll talk about those. Okay, so this is one of the pathways. We'll call them the dorsal column pathway, and involves two tracts, the fasciculus gracilis tract and the fasciculus cuneatus tract. The other ones that we'll talk about is we're going to see two that are found, they're actually found within the lateral white columns, and you'll see in the yellow here and here. On either side up here, this is the anterior spinocerebellar tract. This one is the posterior, and I'm not going to write it out, posterior spinocerebellar tract. So that's going to tell you it's going to go to the cerebellum. These are involved in where it's going to be different. You don't have the decussation like you do with the other ones, is you have the information from the right side of the body is going to be interpreted in the right cerebellum. Information from the left side of the body is going to be interpreted in the left cerebellum, so they're ipsilateral. We also have two that are part of a, one of them is found in the anterior white column, which you see right here, one on either side. This is the anterior spinal thalamic tract. But we have this green one you see on either side here. That is the, we'll do it this one, is the lateral spinal thalamic tract. The lateral spinal thalamic tract is in the lateral white columns. The anterior spinal thalamic tract is in the anterior white columns. There will be decussation with these, but we'll talk about what type of information each of these tracts that we find in the various white columns of the spinal cord. And they are ascending tracts, which means they're carrying information up that spinal cord up to the brain. So we'll talk about specific tracts in class.